the genetic material is DNA was later on proved by A. Hershey and M. Chase in 1952. Viruses which infect bacteria are known as bacteriophages. What they did, they grew these viruses on a medium which is containing, few were grown on a medium containing radioactive phosphorus and other were grown on a medium containing radioactive sulfur. The one which were grown on radioactive phosphorus had radioactive DNA but no radioactive proteins. But the one which was grown on radioactive sulfur, they have radioactive protein but no radioactive DNA. Now the infection is done, that is, they are made to attach on E. coli. Once they are attached, the blending is done in a blender to remove the viral coats, and once the viral coats are removed, centrifugation is done in order to separate the viral parts from the bacteria, and it was found out that the one which was having radioactive DNA is found to be radioactive bacteria, whereas the one which was having radioactive protein is not found to be radioactive bacteria. Hence, it was cleared that DNA is the genetic material which is being transferred from virus to the bacteria, because the proteins, though they were also radioactive, but the infected bacteria is not radioactive. That is, protein is not the genetic material because it is not being transferred from virus to the bacteria. Now, what are the properties of the genetic material? The genetic material should have the property of replication. That is, it can replicate, can bring in the replicas. It should be chemically and structurally stable. It should have scope for evolution. That is, it should not change thoroughly with the life cycle, various stages of life cycle, but it should have scope for slow changes so that evolution can be promoted. It should be heat stable, and as in case of DNA, due to heat, the two strands of DNA may be uncoiled, but as soon as the normal temperature conditions are there, they should refuse to form both the strands.